Hello everyone, a very good evening to all of you. So in this session, I will be discussing a topic from general microbiology, that is staining in microbiology. We have already started staining in the last session, I guess. We have seen the types of the staining and we have covered the important stainings from them. So we have seen there are four types of stains: simple stain, negative stain, impregnation stain and differential stain. The first three type of stains were covered in the last session. So simple stains are the stains which give same color to all the organisms. That's why they are known as simple, because they give same color to all the bacteria. The second was the negative stain. Negative is known as negative because they stain the background, not the organism to be seen for. So you can see the background is black, but you can see the bacteria or the organism which we want to see, they are colorless and they are transparent, they are not stained. So the transparent bacteria will stand out on the black background. So that is the concept of the negative stain. The third stain we have seen is the impregnation stain. Impregnation means layering. So when the bacteria are too thin or the organisms are too thin, the structure we want to see are too thin, we do the layering. The layering is done with the silver. We make them thick so that we can easily see them. That is the concept of impregnation stain. So we have seen these three type of stains: the simple stain, negative stain and impregnation stain. We have already seen their examples. Simple stain have two examples: methylene blue and basic fuchsin. Both of them give same color to all the bacteria. Negative stain, we have seen two examples: India ink and nigrosin. We have already seen that. And impregnation, that is the layering. The layering is done by silver. So silver is the example here. We have already started differential stain in the last session. Differential is known as differential because they give different color to different bacteria. That's why known as differential stains. So in the last session, we have already covered the gram stain. We have seen its principle, interpretation, everything. So we have seen that there are two types of bacteria based on gram staining. Gram positive bacteria, which are violet in the end, and the gram negative bacteria, which are red in the end. So violet and red, the two types of bacteria based on gram staining we have covered. Now it's time to start acid fast stain in this session. So in this session, I will be discussing about the acid fast stain. Very important. So let's start acid fast staining. Acid fast staining, also known as Ziehl-Neelsen staining. Why it is known as Ziehl-Neelsen staining? Although it is discovered by Ehrich, it is modified by Ziehl and Neelsen, the two scientists. So it is modified. It is discovered by Ehrich, but it is modified by Ziehl and Neelsen. That's why it is known as Zn stain. In short, we write as Zn stain. And that is the who does the discovery and who does the modification about the name. Now coming on the method. If you have watched my last lecture, that is on gram stain, I told you there are 4 steps in gram stain. In the similar way, here also in acid fast stain we have 4 steps. So what are the 4 steps? They are written in front of you. So the first step is the primary stain. Here the primary stain is always a red stain, in contrast to gram stain where primary stain was a violet stain. Here any red stain you can use. Usually Carbol, Fuchsin, saffronin, these are the primary stains we use. It should be a red stain, not violet or blue. It should be a red stain. The second step is the mordant. Mordant. Here the mordant is the heat. What is mordant? Mordant is something that fixes the primary stain inside the bacteria, prevent the primary stain to leak out again. So in contrast to gram stain where the mordant we have studied is iodine, here the mordant is heat. Simply heat, no chemical. The third step is the decolorizer. We add decolorizer. So in Zn stain, the decolorizing agent is always H2SO4, sulfuric acid, and it should be 20%. Please concentrate on the concentration, how much we are taking. It is 20% standard. So 20% H2SO4 is the decolorizing agent used in acid fast stain, Zn stain. In contrast to gram stain where we have used either alcohol or acetone. Here we are using sulfuric acid, not alcohol, not acetone. It is the acid. And the last step is the secondary stain. We use secondary stain here, which should be compulsorily a blue stain. It should be a blue stain, not a red. So we have reversed. In contrast to gram stain, we have reversed. You can see we have reversed the colors of the primary and secondary stain. In gram stain, the primary stain was blue or violet and secondary was red. The counter was red. But here in acid fast stain, we have taken the primary stain as a red color and the secondary stain as a blue. Usually we take methylene blue or any blue we can take, right? Now in the end, we will see two type of organisms. There are some organisms which will retain the primary stain. They retain primary stain color. They remain red only, right? They are resisting decolorizer. These are known as acid fast organism. And the organism which lose the primary stain, they are sensitive for decolorizer and they take the color of the secondary stain, right? These are known as non-acid fast organism. Here it is not gram positive, gram negative. So acid fast positive, acid fast negative. We don't use the term positive and negative here. We use acid fast organism and non-acid fast organism. So this is the basic. Let's see the principle and details. Now the best I can explain with this diagram, a beautiful diagram. You know the basic principle, all bacteria are transparent in the world. Do you know this? Yes. So all the bacteria at the beginning are transparent. That's why we are doing the stain. So take the two bacteria. The two bacteria in front of you, both of them are transparent and transparent. Let's do primary stain. Let's start with primary stain, which is a red color stain. So this bacteria also become red and this bacteria also become red, right? And you heat it. You add the mordant, that is heating. So red color become fixed here and red color become fixed here. So step 1 and 2 are done. We have added the primary stain, which is a red stain, Carbol Fuchsin, and we have applied the mordant, that is heat also. Now step number 3. Add 20% H2SO4, which is a decolorizer. Step number 3. Now there are some bacteria which resist decolorizer. Since they are not decolorized by decolorizer, they are resisting it. They remain red only. They remain red only. And there are some bacteria which are sensitive for the decolorizer. They will lose their primary stain because of decolorizer 20% H2SO4. So again they will become transparent. Again, now what makes the difference here? Why some bacteria are resistant and some bacteria are sensitive for 20% H2SO4? Why? Because the difference is the cell wall. Some bacteria have cell wall and in their cell wall they have mycolic acid in their cell wall. This mycolic acid in their cell wall is providing resistance to 20% H2SO4. The bacteria which have mycolic acid in their cell wall, they resist decolorization and they will retain the primary stain. The bacteria which don't have mycolic acid in their cell wall, they will lose the primary stain. They are sensitive to the decolorizer. Now it's time to add the secondary stain. The secondary stain is any of the blue stain, like methylene blue. You add that. Now the bacteria which has retained the primary stain, there is no space inside that bacteria. It cannot take the secondary stain. So it will remain red. It will not allow the secondary stain to come in because there is no space. It is completely full, right? But the bacteria which become transparent, they will take the secondary stain and in the end they will be blue. So you got the two type of bacteria in the end. So in the end, the bacteria which are red in the end, they are known as acid fast organism. Right, and the bacteria which are blue in the end, they are known as non-acid fast organism. I guess the concept is crystal clear to you. So let's revise the two type of organism based on acid fast staining. Zn stain, or also known as acid fast staining. So there are two type of organisms. So acid fast organism and non-acid fast organism. So can you tell me the differences between them? Please try it out. The acid fast organism are the organism which retain the primary stain color, that is they are red in the end. The non-acid fast organism are the organism which take the color of secondary stain, that's why they are blue in the end. Right. Now the acid fast organism, they resist decolorization 20% H2SO4, they resist because they have mycolic acid present in their cell wall. That's why they are resisting. But the non-acid fast, they are sensitive to 20% H2SO4. They lose the primary stain. They are sensitive. They become transparent because they don't have mycolic acid in their cell wall. Right. So that is the difference between them. You got my point? So please understand the difference between them. Let's come ahead. So we have seen the same diagram with a different visibility. You can see on adding primary stain, which is a Carbol Fuchsin, this also become red. this also become red, right? But on adding the decolorizer, which is 20% H2SO4 here, not alcohol, 20% H2SO4, which is acid, some are resisting and remain red because they have mycolic acid in the cell wall, but some are sensitive, so they are losing the red color and they become transparent. That makes the difference. Now add a counter stain, secondary stain, which is a blue stain, that is methylene blue. So the one which retain the primary stain, they still remain red, they don't have space, but the one which become transparent, they will take the secondary stain and they will be blue in the end. So these are acid fast organism. These are non-acid fast organism. I guess the concept is crystal clear to you, and you can tell me the four things now. Tell me the name of primary stain. Say, ma'am, any red stain like Carbol Fuchsin. Tell me the name of the Mordant here. It is heat. Tell me the name of the decolorizer here. It is 20% H2SO4 standard. There are some exceptions. I will tell you exceptions. And tell me the name of the counter stain or secondary stain. It is any of the blue stain like methylene blue usually used. So these are your 4 MCQs, right? You should be able to tell me. So same with a different diagram. Now, in the interpretation, we have already seen the two type of bacteria in the ant: acid fast, non-acid fast. The acid fast retain the primary stain. They are red in color. And the non-acid fast retain the color of the counter stain. That is, they are blue in color. So we have seen that. Can you see a diagram in front of you? In this diagram, can you see the red color rods? So these are the bacteria which are retaining primary stain. They are red. That is, they are retaining the primary stain. So which diagram is it? It is a diagram of acid fast organism. So yes, it is a diagram of Mycobacterium tuberculosis. Can you see the same? These can come in the form of image-based question, right? Or these can, you have to draw these diagram in your university exams if you are a second Prof student. Can you see these? These are the red color rods. Can you appreciate the red color rods? Yes, the red color rods are acid fast organism on a blue background, right? Principle we have already seen. Only one word in principle you have to learn. Some bacteria have mycolic acid in their cell wall, and that mycolic acid resists decolorization. It do not allow decolorization. That's why these are acid fast organism and they retain the primary stain, right? So you can see the mycolic acid in the cell wall. If you compare these bacteria with gram positive and negative bacteria, if you have attended my last lecture, watched my last lecture on gram staining, you may be knowing what makes the difference in the gram positive and gram negative bacteria. Here peptidoglycan content of the cell wall was the main. The bacteria which is having more peptidoglycan content in the cell wall, they were retaining primary stain, these are gram positive. And the bacteria which is having less peptidoglycan content in the cell wall, they were taking the secondary stain and they were gram negative. So in gram positive, gram negative, peptidoglycan content in the cell wall makes the difference. That is the principle. But in acid fast stain, mycolic acid in the cell wall makes the difference. So that is the thing. You got my point. What do you mean by acid fastness? Fastness का क्या मतलब होता है? What is meaning of fastness? It is not fast and slow. It is not related to speed. कि something is fast, something is slow. No. The fastness meaning is resistance. The meaning of the fastness in dictionary, in medical dictionary, fastness means resistance. You know in physics what is resistance? Resistance means opposition. You are opposing something. You don't want that. You oppose that. So that is the meaning of resistance. So here the word is acid fastness. So the bacteria resist acid. Which acid? 20% H2SO4. So 20% H2SO4 is the acid to which the bacteria is resisting. That's why the bacteria are acid fast organism. Acid fast organism and non-acid fast organism. These are the two type of organism I taught you in the end. So acid fast organism, acid fast. So acid is 20% H2SO4. They are fast for that. Means they resist that. So since they resist that, they are resistant to decolorization and they take the primary stain. And non-acid fast means these are not resistance. These are not resistance for the 20% H2SO4. So they are sensitive. So they take the blue stain. So that is the meaning of fastness. Now can you tell me the most important thing? Can you tell me a list of organisms which are acid fast? Can you please tell me the list of the organisms which are acid fast? Yes, I am having a mnemonic for it. So if I ask you this question in your Viva now, you will say only one organism, ma'am, TB, Mycobacterium tuberculosis. You don't know any other name. So these are very important questions for your Viva because the acid fast staining is your practical exercise in microbiology. Along with the theory, it is important in practicals also, right? So see the list. The list is in front of you. The mnemonic is in front of you. No longer separate booking for RIM, RIM, RIM. SIM card. So this is the mnemonic, right? No stands for a bacteria. The name of the bacteria is Nocardia. It is a bacteria. L stands for Legionella. Legionella is also a bacteria. L. Separate ka S stands for Smegma bacteria. It is also a bacteria, right? B. Booking ka B stands for the bacterial spores. Actually not bacteria, the spores of the bacteria. Spores of bacteria, not a bacteria, right? The next is R. Rhodococcus. Rhodococcus, it is a, you can say it is a fungus. Rhodococcus Cryptococcus, it is a fungus. I. Isospora, it is a parasite, right? M. It is Mycobacterium, both Mycobacterium, TB and Leprae. Again, it is a bacteria. SIM ka S is sperm, human male sperm, right? It is not a bacteria, virus, something. It is something extra, you can see. And card ka C is Cryptococcus, also Cyclospora also. So these are the parasites. You got my point? So can you tell me the mnemonic? No longer separate booking. No longer separate booking. RIM, RIM, RIM. SIM, card. So can you tell it again? There are two S in this. No longer separate booking. C. No longer separate booking. RIM, RIM, SIM card. Now try to say the full form with me. Will you try? Say N for Nocardia, L for Legionella, and S for Smegma, and M for Mycobacterium. These are the four bacteria. Legionella, Nocardia, Smegma, and Mycobacterium. Both Mycobacterium, tuberculosis and leprosy. At least these are the bacteria. This is bacterial spores. So this is also bacteria. Actually the spores of the bacteria. Itne to bacteria hai, right? These are the bacteria. Extra mein what you will say? Human sperm. This is extra, not a bacteria, not a fungus, not a parasite. This is something extra, right? So human sperm, you can say that is extra. Now the remaining are fungus or parasite. So let's finish it, fungus and parasite. This is Rhodococcus, this is Isospora, this is Cryptospora and Cyclospora. You got my point? Now if you wish, you can divide them into like how many bacteria, how many parasites, how many others. Others may bacterial spores and sperm, right? How many bacteria? I have told you the names. How many parasites? I told you the names. So in this way, if you want to differentiate like this, you can do this. And if you want to learn like the organisms like this, you can learn this. Can I proceed ahead? Can I? Now, the last thing is the exception or the modification. As I've told you, standard we use 20% H2SO4 as a decolorizer in the process. 20% H2SO4. But there are 4 exceptions. You have to learn the 4 exceptions. For Mycobacterium leprae, we don't use 20%. We use only 5%. For Mycobacterium leprae. For actinomycetes, we don't use 20%. We use 1%. For Nocardia, we use 0.5%. And for Cryptococcus, Isospora, bacterial spores, we use 0.25%. You got my point? So see, standard is 20%, but 5%, 1%, 0.5% and 0.25%, right? So can you tell me the answers? 20% is the standard. It is the standard. But 5% is for Mycobacterium leprae. 1% is for actinomycetes. 0.5% is for Nocardia. And 0.25% is for the parasites, right? Isospora, Cryptospora, for bacterial spores for these. If you don't want to learn all now, it's okay. Learn only Mycobacterium leprae as an exception. It is very important, always asked in the Viva, in your theory papers. So can I say there are 2 types of Mycobacterium. Mycobacterium tuberculosis and Mycobacterium leprae. You know that. For Mycobacterium tuberculosis, we require 20% H2SO4 to decolorize it. But for Mycobacterium leprae, we require only 5% H2SO4 to decolorize it. Yes? Yes or no? Say it. Yes? So 20% and 5%, the 2 type of concentration you can say. So can I say Mycobacterium tuberculosis is more acid fast or Mycobacterium leprae is less acid fast as compared to each other when we compare the 2 mycobacteria? So read this line. Again, a very important line. Mycobacterium leprae is less acid fast as compared to Mycobacterium tuberculosis because it requires 5%, it requires 20% H2SO4. Very important question, right? Now this is all about acid fast staining. Can you compare it with gram staining? Can you compare? Please do the comparison. Please do the comparison. This is gram stain and this is acid fast stain. So can you do the 4 steps comparison? What is the primary stain in gram stain? A violet stain, that is gentian violet. What is the primary stain in acid fast stain? A red stain, that is Carbol Fuchsin. The examples can change. The main is here the stain should be violet, here it should be red, right? The second is the Mordant. What is the Mordant here? Iodine in gram stain. What is the Mordant here? Heat, that is in acid fast stain. Come on decolorizer. The biggest thing is the decolorizer. Here the decolorizer is alcohol or acetone. ethanol. Here the decolorizer is acid, not alcohol, not acetone. It is a 20% H2SO4, that is an acid. Come on, secondary stain. The secondary stain is always here a red stain, and here it is a blue stain. You can see here primary stain is violet or blue and secondary is red, and here in acid fast it is reverse. The primary is red and the secondary is blue. So you can see the change. Now what is the interpretation? Come on, the interpretation of both of them. Come on, the interpretation of both of them. In gram stain, some organisms are violet in the end, they take primary stain. So that's why they are gram positive. Some take the stain of the secondary stain, that is they are red in the end, they are gram negative. So the violet one are gram positive and the red one are gram negative. That is the interpretation. What is the interpretation in acid fast? In acid fast, some retain the primary stain, so they are red in color. They are acid fast organism, right? The acid fast are the red in color, and some take the secondary stain, right? So they are blue in color. So non-acid fast are blue in color. You can see the difference, you can make it out. Now if any of the, no, can you, the last thing, can you tell me the change, the difference in the principle in both of them? Why these changes are there? Here between two bacteria, the difference in color, the difference is due to the peptidoglycan content. The peptidoglycan content is more, so they will retain the primary stain. They are gram positive. And if peptidoglycan content is less, they will lose the primary stain and take the secondary stain. They are gram negative. So peptidoglycan is the principle. And here the principle is mycolic acid in the cell wall. If mycolic acid is present, they will take primary stain, that is red. And if it is absent, they will take the secondary stain, that is blue. So here mycolic acid in the cell wall. Both are cell wall, but the content is different. I cannot simplify more than this. Now the next will be the example I will be telling you. Can you tell me the examples on the basis of gram stain, how we have divided? See the two stains, compare, very good diagram. Can you do the comparison? Compare. See gram positive organism, they are violet. See gram negative, they are red. Violet, red. See these are acid fast organism, they are red. Non-acid fast, I have not put the diagram. So that is the diagrams. Now gram staining, based on gram staining, we divide the organism into 4 categories. In the last session, I told you the example. Gram positive cocci, gram negative cocci, gram positive bacilli, and gram negative bacilli. We have seen the examples. If you want me to say it again, gram positive cocci are Staphylococcus streptococcus, only 2. Gram negative cocci, again 2, Neisseria and Moraxella. Right? Gram positive bacilli, they are 5, A, B, C, D, L. Actinomycetes, Bacillus, Clostridium, Diphtheria, Listeria. Right? These 5. And gram negative bacilli remaining all. Don't learn the list. Remaining all. So that is gram staining. Based on gram staining, we divide like this. Based on acid fast staining, only these organisms are acid fast. Remaining all, jitne bhi aur bach gaye, remaining all are non-acid fast. So don't learn the list of non-acid fast. Acid fast itne hi hai. You know the mnemonic. No longer separate booking for RIM SIM card. Right? So that is the thing. Now, before I would like to ask you 2 questions here. Before that, let me tell you, if in your university exam, gram stain or acid fast or Ziehl-Neelsen stain is coming, what you have to write? The first thing you have to write is the method. The 4 steps here, the 4 steps here. Then you have to write the principle. What makes the difference? The answer here will be the peptidoglycan content in the cell wall. The answer here will be mycolic acid content in the cell wall. You have to describe it in points how it makes the difference. Then you have to draw the diagrams of the 2, positive, negative here. You have to draw the diagrams here, acid fast, non-acid fast. Then you have to write the interpretation here. So gram positive are violet, gram negative are red. You have to write the interpretation here. Acid fast are red, non-acid fast are blue. And then in the end, you have to write the examples here. You have to write the examples here. So if you are writing in these headings now, you will get full marks. So if you are getting it for your 5 marks, you have to be prepared for that like this. Right? So give me a thumbs up, everyone. Right? Now I would like to ask you 2 questions here. If you have attended my lecture attentively, please answer these 2 questions. Please. The question number 1 is in front of you. Decolorization in Zn stain. If I'm not asking specifically for any bacteria, I want to ask you standard. So in this question, I want to ask you standard. What is the standard decolorizer we are using? I'm not mentioning any name of the particular bacteria now. So is it 1% sulfuric acid, 5% sulfuric acid, 10% sulfuric acid, or 20%? ABCD, what is your answer? Please drop your answer in the comment box right now. Number 1. Number 2 question is in front of you. Can you tell me the decolorization of Zaden staining, the same question, but I am asking specifically for Mycobacterium leprae. So I have mentioned the name. Have you understood the difference between the two questions? Here I have not mentioned the name of any bacteria. So I want to ask you standard. Here I have mentioned the name of a specific bacteria, Mycobacterium leprae. So the same question. Can you tell me the concentration? Is it 5%, 1%, 0.5% or 0.25%? What is the answer? So please drop the answer in the comment box. This is question number 1. This is question number 2. Please, I will tell you the answer in the comment box only. So after this, I would like to tell you, if you have any doubt in this session or any session of pathology, pharmacology, microbiology, or whatever I am teaching you, you are free to ask your doubts along with your name, along with your university of your exam, along with your probable exam dates if you are a second prof student, right? And what exam you are targeting exactly. For you are watching my videos for which exam? You are watching for university exam, or you are watching for some competitive exam like Neet PG, Next, INI-CT, FMGE, whatever, USMLE, what you are preparing for, currently what you are targeting. So this is my contact number. It's 9833032948. You can connect to me either on WhatsApp or on Telegram. Kindly don't call, just drop a message. So whenever I will get time, I will definitely respond you. Okay. And before leaving a very important motivation. So every day I give you a small motivation, right? We all have 24 hours, right? So everyone have 24 hours. But in these 24 hours, some dreamer just consume in dreaming, and some dreamer consume those 24 hours in working. So you are just dreaming, or you are working actually in 24 hours. We all have 24 hours, of course. So some only dream, one day this will happen, I will become a great doctor, I will pass my exams with rank 1, single digit rank, I will take this branch, I will do that, I will be having a good house, good money, good partner, good everything. You are just dreaming, but you should start working for that. Just dreaming will not fulfill your dreams. If you want to fulfill your dreams, if you want your dreams should come real one day, you should start working. You should be serious now. Now you all are a medical students. You are not normal, some layman. You are a medical student. One day you are going to become doctor, right? It's a big honor to become a doctor, I'm telling you. People rely on you. People trust on you. Millions of tons of people trust on you. You are a doctor. They consider you like God. Oh my God. When people trust me like so much now, I got afraid from inside. Why they are trusting me like so much? They just, they just अपने हाथ जोड़ के कहते हैं, doctor, you are a doctor, just save the life of my dear one and just save my life. So I feel so like they are trusting on me. They are trusting. I cannot break the trust of anyone, right? They are trusting on me. They are trusting on my knowledge, on my study. I have to treat them. I have to apply my knowledge, whatever I have studied in my MBBS and post-graduation, for diagnosing, treating the patient accurately. I should guide them properly. So that is my moral responsibility, and yours moral responsibility as well, I guess. So you should stop dreaming, just dreaming. After dreaming, dream it. I'm not saying no. At least in a day, once or twice, you should dream. Dream means dream with your open eyes, not sleeping. So just dream your open eyes that one day this will happen. So whatever is your dream, start working for that. Start working hard. You should become serious in life now, right? बहुत हो गया, अब serious होने का time आ गया है. So serious हो जाओ, अच्छे से पढ़ाई करो, अच्छे से यूनिवर्सिटी एग्जाम में रैंक लाओ, अच्छे से कॉम्पिटिटिव एग्जाम में रैंक लाओ, बढ़िया से किसी ब्रांच में, अपनी मनपसंद ब्रांच में, बढ़िया से इंस्टीट्यूट से अपना पोस्ट ग्रेजुएशन करो, और एक डिग्निटी के साथ बाहर आओ, एक प्रेस्टीज के साथ, एक ऑनर के साथ बाहर आओ, तो लाइफ में मजा है, तो मजा है. पीपल विल सी यू विद दैट ऑनर. So that is the thing, right? So thank you very much for being with me. If you like the lecture, don't forget to click on the like button, and please share the link with all your friends, colleagues, everyone, all the medicos, I must say, throughout the globe, in your contact in any country throughout the globe. Please share the link with all medicos. It will be useful for them, right? So thank you very much, and don't forget to subscribe my YouTube channel and press the bell icon if you want the notifications in future. Thank you. See you. Bye bye. Study hard. All the best.